Good morning. Whew. I just woke up. It's 6 a.m. I sleep under my desk. All teachers sleep under their desks. We live here. Don't you know that? I got my water. I don't plan on eating today, though. That's okay. I'm not really hungry. This is Chapter 8, Part 2. Enzymes and all their functionality. So let's talk. Specifically, we introduced metabolism in the last section. And now we need to talk about how enzymes regulate these metabolic reactions. And largely they do so by lowering energy barriers. Okay, a catalyst is something that speeds up a reaction, and it does so without being screwed. It doesn't get messed up. An enzyme is a very specific catalyst. It's a catalytic protein that functions in cellular type environments. So an enzyme is a specific type of catalyst. Okay, so there's a series of chemical reactions that we're going to show you later. These can involve bond breaking or bond forming. That's completely specific to the reaction that's going on. Here's an example of a chemical reaction. Okay, sucrose being broken down by the enzyme sucrase that you see here, forming glucose and fructose. Okay, this is a hydrolysis because water is causing the splitting. This chemical reaction is regulated by that enzyme sucrase. Okay, now by itself, sucrose could break down, although it might take a very long time. It takes a very long time because of its energy of activation, or activation energy that's in the way. This is the initial amount of energy needed to start the chemical reaction. Often supplied in the form of heat from something within the system. Okay, so here's an example from an exergonic reaction. Reactants have more energy than the product, so it's a downhill, if you like. But there is this temporary transition state that must be overcome because of this activation energy. So enzymes, you'll see, help to lower this activation energy. Okay, as you see here in red with an enzyme, it lowered that energy, so it's easier. Imagine if you were walking up that hill. Okay, the smaller or lower the hill, the easier it is to get over to the products. Okay, delta G specifically is unaffected by the enzyme. There is no change in free energy. There's only a change in the activation energy needed to be overcome to make the reaction go to completion. Now, an enzyme is always going to act on a substrate. That's the reactant or the thing that's being acted upon. The enzyme itself is going to bind to the substrate and form this big complex that we call the enzyme-substrate complex that you see here. Okay, the substrate is going to bind in the active site of the enzyme. That's the one region where it fits perfectly, and they're highly, highly specific. And it creates an induced fit of the substrate, as we say. It's essentially here you have the enzyme-substrate complex. This enzyme is now going to act on that substrate. Okay. This is showing you the enzyme-substrate complex. It alters the substrates, the reactants in this case. We have our induced fit, okay? And the substrate is now converted into these products. What you'll notice is that while the enzyme may change shape, it never alters itself, okay? It can break or form bonds within the products, but the enzyme stays intact. Enzymes can get old over time, so to speak. We won't talk details about that right now, but in general, they stay functional, and only the substrate is altered or changed. Now, enzymes don't work perfectly everywhere. There's different environments that they work, but they all work by lowering the activation energy, and that is lowered by orienting the substrates properly so that they can be properly separated or bound. So if I need to bind my pointer finger to my thumb, if the substrate is oriented like this, it's going to be very difficult to bind. But if an enzyme shifts and rearranges this, well, now these substrates are oriented correctly and I can form that bond. I can strain substrate bonds. So if I have a substrate that's shaped like this, okay, I'm not providing very much strain to break that bond. But if I now, with an enzyme, shift and strain this substrate like this, now it's easier to break that bond. So straining substrate bonds, providing a favorable microenvironment, so altering the environmental conditions, making it more hydrophilic or hydrophobic, for example. Acid or base to affect the strain or the orientation of the substrate, allowing it to be affected. And then covalently bonding to the substrate directly can alter the substrate shape and allow it to be to minimize the activation energy. So what kind of environmental factors can affect it? Temperature, okay, and pH. Here we see human enzyme. This is the optimal temperature at which this enzyme works, about 37 degrees. That's Celsius. That's the for this specific human enzyme. I don't know which one they're referring to. Versus a heat-tolerant bacteria, their ideal enzymatic temperature is up here at 77. Okay, so they like it hot. Some like it hot. You don't know who Marilyn Monroe is, either do I. But anyways, pH is the same thing. So a stomach enzyme called pepsin loves a pH of 2. Okay, if the pH is off of 2, it doesn't function as well. We'll talk later about why that is. Optimal pH for trypsin, which is an intestinal enzyme, 2 different environments. In the stomach, it's very acidic. In the intestine, it's more basic. It likes a pH of 8. Okay, both of them actually digest protein in different environments. Now, enzymes need some help sometimes. That comes in the form of cofactors and coenzymes. Cofactors are non-protein helpers. Things like metallic ions, for example, magnesium could be a cofactor. When bound, when part of the enzyme, it helps the enzyme maintain its shape or its specific form that enhances its functionality. Coenzymes, the same type of thing, but they're organic type cofactors, okay? Such as oxygens or what have you, okay? Now, the enzyme itself, because it has a specific active site, sometimes things can get in the way. And this is a large basis for drug production, actually, where we have competitive inhibitors, where we want to have something get in the way of the active site so the enzyme can't bind with the substrate. Here, this competitive inhibitor is in the way, and as a result, the substrate here, this aqua colored thing, can't bind into the active site. It's competitive inhibition. Non-competitive inhibitors bind away from the active site, and the result of their binding causes a change in the shape of the active site of the enzyme, and as a result, it can't bind or reduces the ability to bind to the substrate. Now, these various pathways, the metabolic pathways that we've talked about, and we'll talk about more in various other chapters, all are regulated by enzymes. And it's important. Every single step, as opposed to jumping from the very beginning of the pathway to the end, there's many, many steps in between, as I mentioned previously, because we can upregulate and downregulate at every one of those steps, make them go faster, slower, block them, turn them on, and so on. It has to be super tightly regulated so things happen properly. So here we have allosteric regulation, a really type of common regulation. And this is any case in which a protein's function at one site is affected by a molecule's binding at another. We just call it a regulatory molecule. They can be allosteric activators or inhibitors, and many enzymes are allosterically regulated. Here, for example, is an enzyme. It has an inactive form, which is non-functional because the shape of the active site can't bind the substrate. And here's the active form, and they oscillate back and forth between the two. If we want this enzyme to function, to work, we can bind an allosteric activator to it, which enhances the active form and maintains it in this stabilized fashion so that it can continue working. And then in order to turn the enzyme off, we would remove the allosteric activator and it would go back to its inactive form. Now, on the other hand, if we had an enzyme that we wanted to turn off, we could bind the inhibitor to it and turn off this enzyme, so now it's allosterically inhibited. All the active sites don't function properly. Now, cooperativity is a very specific type of allosteric regulation, where a substrate binds to one of the subunits of this quaternary structure, and by doing so, it locks in the active form of all the other subunits. So now all of them have this enhanced binding. This cooperativity is actually seen in hemoglobin in your bloodstream. Hemoglobin has 4 different subunits, and the more oxygen that's bound to those subunits, the stronger the affinity of the other subunits are to oxygen. So the more oxygen that's bound, the easier it is to bind more oxygens, which means essentially that usually you don't find hemoglobin with just one subunit binding oxygen. It's kind of like all or none because of that cooperativity. Now, pathways can be altered not only by, or specifically when they are, when you target each specific enzyme, and sometimes we have too much of an end product in a metabolic pathway that we don't want to make anymore. In this case, for example, we are trying to convert threonine, an amino acid, to isoleucine. Various enzymes in the pathway affect that and all those different steps. If we have too much isoleucine, we can feedback or negatively inhibit, through negative feedback, the isoleucine will act as an allosteric inhibitor and bind to the enzyme. This enzyme is called threonine deaminase. You don't have to know that, of course. But when bound, this enzyme, this threonine deaminase, cannot bind to threonine. Notice the active site has changed, and as a result, you won't make any more isoleucine. You've turned the pathway off. And the same goes when this unbinds, it now turns the pathway on, because there's not enough isoleucine. Okay? Where do we find all these enzymes? Well, in the cell, they're found all in different places. They're grouped into complexes, groups of enzymes, as we'll see in DNA replication at a much later date. They can be incorporated into membranes and housed inside there, largely because if I have an enzyme that is going to degrade this pen and break it apart, well, I don't want to put this enzyme in an area where there's lots of pens that I want to keep intact. I want to hide that enzyme away behind a membrane, and only when I'm ready to use it, I release it out of that compartment and expose all these pens to that enzyme to break it apart. But otherwise, I don't want to constantly degrade these things. I need them to write on the board, right? An example of where they might be kept is inside the mitochondria, for example, or inside a lysosome, or inside the Golgi, all these various organelles that you will learn in Chapter 6. And on that note, I am...